Hi, everyone. So now we're going to move into our second chunk of 15-minute talks. And the first speaker that we're going to have is Audrey Brown from the University of Virginia. So Audrey, do you want to go ahead and share your screen? And while she is doing that, just give a brief introduction of Audrey. So Audrey received her undergraduate degree from Western Kentucky University before moving to the graduate school at University of Virginia, where she's in the final year of her PhD in Jennifer Guler's lab. So she studies how changes in the parasite environment can affect drug sensitivity and pathogenicity. So take it away, Audrey.
Lovely. Can you all hear me and see my screen just fine?
Yes, we can.
Great. So yes, as Tori mentioned, my name is Audrey Brown. I'm in Jenny Guler's lab at UVA, and today I'm talking about how broad nutrient stress can actually increase P. falciparum survival of drug artemisinin. But after I submitted this abstract, I realized that I actually missed an opportunity to instead call this talk, "What Doesn't Kill You Makes You Stronger." And by that, I mean that mild stress can actually be beneficial as organisms adapt to that environmental stress and potentially even overcompensate. So an example of this in humans can be exercise. So regular exercise actually makes you more fit, as evidenced here by these nice, well-adapted stock photo models. However, this type of stress can be taken too far. So in our exercise example, if you exercise overly much, you can actually lead to excess inflammation or even injury, as shown here by this less happy stock photo model. But what does this mean in the context of microbes like P. falciparum? So we know that changes in metabolism and changes particularly in the ability to respond to nutrient stress can actually affect drug sensitivity in P. falciparum. So we can imagine a scenario in which sort of mild stress can alter metabolism, potentially by increasing stress responsive pathways. And then those already activated stress responsive pathways are going to be beneficial in the face of a subsequent stressor. In the case of this presentation, that's drug treatment. So this sort of phenomenon is what we're calling metabolic priming, leading to our hypothesis that we believe metabolic priming can lead to increased survival of drug tolerance in P. falciparum. Specifically, we're interested in the context of nutrient stress. In regards to that, because of this previous work that I mentioned, some of which are cited below, and also because we know that nutrient stress is something that varies among different patients who actually have clinical infections. So our paradigm for testing this hypothesis is shown here. So our parasites are put into one of our defined low nutrient media conditions for 36 to 72 hours. And so we've developed a few different low nutrient conditions based on using growth and viability data to ensure that we are having a stress effect on the parasites, but not so harsh of an effect that we're actually killing off large sections of the population, that we're in that what we think to be the hormetic range. We then treat parasites with a 6-hour pulse of 200 nanomolar of dihydroartemisinin, shown here abbreviated as DHA. We then wash off the drug and put all parasites back in regular rPMI plus Albamax and monitor growth and proliferation for about 10 days. And our prediction from this paradigm is that parasites that receive this metabolic priming step in the low nutrient upstream DHA will actually have superior recovery, will have this hormetic effect whenever they're treated with DHA. So this is what these data look like, and we actually do see just that. So on these graphs, on the x-axis, we have time, where day 0 represents the day of that drug pulse, and then we're looking at growth for 10 days following that. And our primary metric of outcome is actually looking at this difference in growth at day 10. So sort of the fold and percent increase that we see in those primed parasites of proliferation. And we do see that consistently between different types of stressors, that the primed parasites actually recover and grow better than their non-primed counterparts in either low hypoxanthine media, complete thiamine dropout media, thiamine being also known as vitamin B1, and also in reduced glucose media. So here we have shown 3 specific stressors where we're either reducing or dropping out a specific metabolite. But we've also done this using something called human plasma-like media as our priming stress in place of rPMI. So using human plasma-like media as a base plus Albamax being the priming condition versus the non-prime condition being our standard control condition of rPMI plus Albamax. So the human plasma-like media contains nutrient levels that are roughly akin to what's in the human bloodstream, which in many cases is much lower than what we see in RPMI. So here's just a few relevant metabolites pulled out down here on the bottom right. And you can see that some metabolites are in equivalent concentrations between these base media, but many of them are much lower in human plasma-like media relative to their RPMI counterparts. So we think of this as being sort of an overall decrease in many different metabolites, potentially leading to nutrient stress. I think it's important to note that these differences that we see at day 10 in proliferation are dependent on that drug pulse. So for example, if you take primed and non-primed parasites and you do not apply the drug pulse, if you just put them back in regular media and watch them grow, they grow at exactly the same rate, or sometimes the primed ones actually grow a little bit worse. So here that's represented by these two solid overlapping lines. It's only whenever you add that drug pressure that you get that discrepancy shown there in those dashed lines. So this data is shown for DHA. Whether this type of phenotype is limited to DHA or can be found in other drugs is yet to be known. So that's something that we're interested in exploring in the future. So our primary metric of output on these types of assays is this day 10 difference in growth. And what we wanted to know was what factors affect this number. So we pulled out different attributes that vary between experiments and put them into a linear model to see what affects this outcome. And we found two things of interest. So one being whether or not the parasites were treated with drug. This makes sense based on the previous slide I showed you, where if you don't pulse with drug, you don't get this hormetic effect in difference in growth. What we also found was that the percentage of rings at the end of priming, right before they received the drug treatment, was also affecting this factor. And by that, I don't mean that within an experiment, the primed and non-primed parasites had different levels of rings, because they don't. They're pretty consistent within an experiment. The priming treatment doesn't seem to skew staging. But between experiments where the parasites generally had lower levels of rings had a lower magnitude of day 10 survival relative to ones that had a higher percentage of rings. This is just two examples of experiments. So this led us to believe that the ring stages could be primarily mediating this impact. And so we decided to proceed by a ring stage survival assay. So this is a standardized assay for assessing DHA tolerance and resistance in Plasmodium, specifically on rings within 0 to 3 hours post-invasion of red blood cells. So here I have these data shown with 200 nanomolar DHA, which is what was used in these previous graphs, also with 700 nanomolar DHA, which is the more conventional concentration used for the assay. So here you see that the primed do in fact grow about 2 to 4-fold better in these assays. However, whenever we did the same thing that we're calling a trophozoite survival assay on parasites 24 to 27 hours post-invasion, we actually don't see that difference anymore. That difference is ablated. So this is giving us further evidence that this benefit from metabolic priming is likely mediated by rings. So we have this set of metabolic stressors, some of which are individual decreases or dropouts of metabolites, and one of which is sort of a broad overall reduction in metabolites. But they all show this same hormetic effect. So that's why we were very interested whenever we tried this with isoleucine reduction and did not see the same effect. So by reducing isoleucine in the media, we actually don't see any benefit of priming. The primed parasites actually grew slightly worse. And so to understand why this condition was different than the other conditions, we shifted from doing these phenotypic growth assays into investigating the molecular mechanisms. Starting with looking at something called EIF2 Alpha. This is eukaryotic initiation factor 2 alpha. It is required for translation initiation in the parasites, and phosphorylation turns this protein off, so thereby conserving resources, limiting translation. And it does actually allow for some specific translation of stress responsive transcripts. So this EIF2 alpha phosphorylation is known to occur in response to nutritional stress. It is also known to actually be protective for DHA treatment, leading us to hypothesize that this phosphorylation is actually being induced during our metabolic priming step, and then it's subsequently allowing for increased survival upon DHA treatment. So the first experimental step to assessing this was to actually quantify phosphorylation in our prime samples. So this is quantification of Western blotting for the samples that we have done so far. And these samples were taken prior to drug treatment. So this is just the effect of the metabolic priming alone. And what we see is that the hypoxanthine deprivation, thiamine, and reduced glucose do actually seem to have an effect, an increase in phosphorylation relative to their non-primed control. However, we don't see that in isoleucine, which is further interesting because other groups have seen a phosphorylation induced by low isoleucine conditions. So we believe the difference is the amount of isoleucine and the amount of time. So for example, you can see a paper referenced here that did see EIF2 alpha phosphorylation, but they used much lower concentration of isoleucine and for a much shorter period of time, a sort of harsher treatment. So it's not that isoleucine cannot induce phosphorylation and maybe subsequently could not have a hormetic effect in the proper range. It's that our conditions aren't permissive to it. But this did allow us to see something very interesting. If you look at the pattern of increases in phosphorylation and you look at the pattern of increases in growth at day 10, they sort of track with each other. And so we decided to put these against each other in a correlation, just as preliminary data exploration. And we see that this increase in phosphorylation, this increase in survival have a very striking correlation, indicating to us that we are likely on the right track and the EIF2 alpha is likely somehow involved in this pro-survival hormesis phenotype. The next thing that we're going to do is investigate this further by using an episomally expressed EIF2 alpha dominant negative construct. So this has a mutation in it that will not allow it to be turned off, driving continual transcription and translation through the cell. And so we believe that this nutritional priming will no longer lead to this hormetic effect whenever this dominant negative is expressed in these cells. We also plan to do RNA-seq of primed parasites to look for stress responsive changes, particularly looking if any genes pop out that are known to be associated from the literature with DHA survival or tolerance. So what does this all mean for malaria and malaria treatment? So we likely think that nutrient priming likely occurs much more frequently in vivo than in vitro. So for example, this could occur from overall reduction in metabolites, such as in our HPLM media, or this could be more specific, such as malaria-induced hypoglycemia. So it's possible that our parasites in vitro are actually in the A part of this curve, whereas in the context of an actual infection, our parasites are in this B part of the curve, surviving a bit better than we think that they otherwise would. So if this is the case, this is something that could be leveraged, potentially developing small molecules to target the stress response to be used as an adjuvant with artemisinin in order to continue to increase its efficacy, even as tolerance to artemisinin spreads throughout the world. With that, I would like to thank my lab and my funding sources, particularly thanking my PI and Michelle Worthen, who contributed data to this presentation. And with that, if we have time, I will take questions.
Thank you, Audrey. We are running a little bit short on time. So I'm just going to ask one quick question. So you mentioned this EIF2A phosphorylation. Do you know if that same kind of process is used or is seen with bacterial persistence? Like how relatable is this to other microorganisms?
So it's very relatable to other eukaryotic cells, but wouldn't be relatable to bacteria. But this EIF2 alpha phosphorylation in response to stress does occur broadly across the tree of life in eukaryotic cells, but is not, it sort of can produce a similar phenotype to like bacterial persistence, but it's not the same mechanism.